This is a production of Cornell University.
All right, good afternoon, everyone. So I'll get straight into it. So here in the central region of New York, we have grapevines that are cultivated around the Finger Lakes. And this is because of some special characteristics of the area. And you can see here in this map that you're seeing, this is the USDA hardiness zone map. And you can see in green where the area with a milder climate extends from the Great Lakes further south because of the Finger Lakes. So it is because of these characteristics that we are able to cultivate grapevines of the vinifera kind around here. And you can see that if you look at the map of where we have wineries in the state of New York, you'll see that they are located where those green areas in the map to the left is. So there's climate characteristics that are that become milder because of these large bodies of water that we see here in the western part of New York. And but still, there's some years where the temperatures drop. They are too low, so we are studying cold hardiness in grapes to try to understand what are the characteristics in the wild grapevines that make them survive even further north from here, and that the cultivated grapevines are not, that they don't have. So here, it's just to show you there's different characteristics for each of the Finger Lakes. So it's a combination of the geography and the depth of the lakes and the surface area of them that prevents them from, in most winters, from freezing. And because of that, they have they buffer especially the low temperatures in the area. And then you might tell me, but global warming, we're increasing the temperatures. Why is your study relevant? Why do we need to understand cold hardiness if we're not going to need it in a near future? And then I'll tell you, here in these maps, we see that the winter temperatures are increasing, and even the extremes in temperature are going up. And this is basically based on that the low temperatures are rising. It's not the highs, but the low temperatures. And the reason why it's still relevant is because of this type of phenomenon that still is going to occur. The polar vortexes that we've been seeing with greater frequency, it's not just because we came up with a name for them, but they actually have increased in frequency lately. Because as the air warms in the southern regions, it decreases the strength of that jet stream. And because of that, it's able to move further south, and then the Arctic air moves further south. So we're still going to see low extremes in temperature, even though the winter temperatures are milder. And hopefully by the end of this presentation, you'll able to make your own conclusions in how, because of how grapevines are perceiving temperatures, they might still be affected by these events. So because the seminar was titled Grapevine Survival, Winter Survival Guide, I decided to talk about fight or flight responses in the grapevines. And so this is the sort of thing, like a general grapevine that you have in the late summer with the fruit. The first thing that leaves the grapevine are the fruit that we're picking. And then as the days become shorter and the quality of light changes, you start getting the dormancy established, so you get into fall. And once those leaves are in the flight response, so they're going to get away, but part of that plant is still going to be there. So you have all the dormant structures that have to resist the winter temperatures. So I'm going to talk about, especially today, what I'm going to talk about is how the buds of the plants are going to survive the winter and how they respond to temperature. Some of the things that I'll be talking about: endodormancy. What is endodormancy? If you cut a bud off of a plant and you try to grow it very early in the winter, when it hasn't seen or felt any cold, it's basically not going to grow. You can see here this cutting, it grows roots, but it's not developing that bud. So you're not seeing bud break in here. It's not growing. Whereas when they're eco-dormant, you actually see the buds start to grow very quickly. And if you compare the sizes of the roots, you'll see that this started growing much earlier than the roots. And so the transition from this endodormant state, which is a non-responsive state, into an eco-dormant state where if you give enough temperatures, they'll start growing, is understood as being a response to chill accumulation. So the plants have a clock in which they're counting how many hours they're spending in low temperatures, typically calculated between 0 and 11 Celsius, somewhere in that range, depending on what model you're using. And so if we look at the map for chilling accumulation here in the US, you'll see that if you go further north, you have more chilling compared to the south. But if you look at the Midwest region, you'll see that it actually has less chilling, or it's comparable to the southern regions. That's because they spend so much time in the below freezing temperatures during midwinter that they really don't accumulate chilling. So the plants don't count that into their clock. So you have that in mid to high latitudes is really where you have most of your chill accumulation. And then if you go further north, you don't see that anymore. So here, just so you can see some data, and it depends on the year also that you're looking at. So this is a warm year, and you see that here in New York and Missouri, you're going to 1,600 hours, and in South Dakota, you only get 1,000. Whereas if we have a very cold winter like 2013, 2014, we actually have less chilling accumulated here in the New York region, much closer to what South Dakota gets. So it really fluctuates, the responses of the plants fluctuate from winter to winter. I'll also be talking a lot about differential thermal analysis, or LTEs are the term that you're gonna hear me say. This is the analysis that we do to evaluate the cold hardiness of these buds. So we cut them off of the plant. And we're careful to leave some of the bud cushion, because this is important for them to still have this phenotype that we're measuring. We put them in these thermoelectric modules that are that measure changes in voltage based on heat. And so we have these large plates, and we put them in a programmable freezer, which is going to lower the temperature until -40 Celsius. And then we get this record, and here the axis for temperature is flipped, so the more negative temperatures are to the right. And you'll see that we see a high temperature exotherm, or HTE, which is non-lethal. And then we have a low temperature exotherm, which is where the buds are being killed by the temperature. And here you see 5 peaks from 5 different buds. And so we are able to record the temperatures where these buds are killed. This is based on a supercooling ability that the buds have inside of them. So the water is actually able to drop below its freezing point without forming ice crystals. And for you to understand what this process is, I make these videos every single winter. I have a ton of them to show what this looks like if you just look at water. So you'll see that that water is liquid, but once you shake it, it actually crystallizes. So that water was super cooled, and then once I shook it, it was actually able to form the ice crystals. It doesn't completely freeze, because as it's freezing, there's heat being released, so it's only partially becoming solid. So we measure this throughout the winter, and you'll see that this curve has a generally, sorry, I'm trying to get rid of that green line that was there. I can't, so I'll just keep going. So you see that it has a general U shape that follows the air temperature, which is in black. So in the bottom, you see the cold hardiness for Cabernet Sauvignon and Riesling. And so we have what we think about as 3 phases. So you have an acclimation phase in which the buds are gaining hardiness early in the winter. You have a maintenance phase in midwinter where they're not varying that much. And then in the spring, they start losing their hardiness, so they are deacclimating. And you'll see here that there's one point where the air temperature actually goes below what the cold hardiness is for those buds. Actually, there's 2 points where it gets very close. And what we see when we take those buds from the field is that we actually see damage in them. So the bud anatomy in vinifera is that you have within a bud, it's a compound bud, so you have 3 smaller buds. And the primary is the one that has the flower. So that's the one that's going to give you the grapes. And when we see that peak exotherm, or when we see the temperature dropping below where the cold hardiness was, we see this damage here where there's a browning in the primary bud. Whereas the secondary and tertiary are still green in this case. So they are able to, the plant will still be able to grow after this damage, but it won't have flowers or grapes from this bud at least. When we look at 2 separate winters, we see that they vary across winters in how they gain hardiness and how they lose hardiness. But there's about a constant in the maximum hardiness that they're able to get, and which for a vinifera is about -25 Celsius. So if you have temperatures constantly dropping below that, you're not going to be able to cultivate vinifera grapevines, which is where the ones we get most of our wine from. So you might say it responds to temperature, so why can't we predict this? And well, we can in a way. There was a model that was developed in Washington State using field data, and it works very well for them. But when we use this model to predict our cold hardiness, it doesn't do a very good job. So in green, you see our data points of our measured cold hardiness. And in orange, you'll see what the model predicted. So really what we see is that there's an over-prediction of hardiness in the acclimation phase and an over-prediction of hardiness in the deacclimation phase. And so really the acclimation phase for us is not that much of a problem, but when we look at the deacclimation, here in the Northeast, our coldest temperatures of the year are really in the last portion of the winter. So if we have warm temperatures midwinter and they start losing their hardiness, it's very concerning that the model doesn't predict that for us because of this shift in when the coldest temperatures of the year are here. Sorry, the darkest colors are later in the winter. And so we decided to just test different temperatures to see how they acclimated and just fix the rates that they were given for each one of the temperatures. And it sounded like a very simple idea. So we took buds from the field and we made these single node cuttings and we exposed them to different temperatures, 2, 4, 7, 11, and 22 Celsius. And we really expected them to gain hardiness at least in the 2, 4, 7, but the model actually predicts that at 11, it's still gaining hardiness. At 22, we didn't expect them to gain anything. When we looked at the data, for 5 weeks, they did not gain any hardiness in those chambers. But when we looked at the field, they were, at the same time, they were gaining hardiness. And so we went to see why this was happening. Looking at the field temperatures, yes?
Were those rooted cuttings or what?
No, they were not rooted cuttings. They're just the buds and single node cuttings without a root.
And then held at these temperatures for 5 weeks.
Yep. Mm-hmm.
Okay.
Yes. So when we look at the temperatures in the field, we see this, and comparing to the temperatures that we had in our growth chambers, you'll see that the blue line, which is 2 Celsius, is really close to the minimum temperatures that these buds were experiencing out in the field, except for really the later portion here of that period. And it didn't really make any sense that at least in the 2, they should be gaining some hardiness. If we look at the average the daily average temperature instead of the hourly data, you'll see that really for most of the period, it is above that 2 Celsius. And the average for the whole period is actually around 7. So we really expected them to gain hardiness. But there was something to learn about this, that even though they were touching that 2 degrees line, it was cycling. There was some temperature amplitude going on. And so really what I did was I tried to search things related to how temperature cycling affected plants. And I came across an entomology paper that said, the title was something like Effects of Neglecting Temperature Amplitude in Insects. And even though the results were not relevant to this, I liked the title that was The Effects of Neglecting. And this is exactly what we were doing. We were neglecting the temperature amplitude that they experience in the field and just going with constant temperatures. So when we decided to expose them to a cycling chamber. So we took buds from 2, 7, and 11 degrees Celsius, and we moved them into a chamber that was cycling from 1.5 Celsius up to 12.5, so that the average temperature was still 7 Celsius, but they were going very close to what the 2 was and a little bit above what 11 was. When we looked at these buds that were now experiencing cycling temperatures, we saw that after we moved them into the cycling, they gained hardiness. So really, even in the 2 Celsius, which was essentially only experiencing temperatures higher than what it had seen before, they did gain hardiness in that cycling chamber. So we came up with a term that took into account daily variations in temperature, which is a sigma T is what we called, that would perhaps fix our model there. We also did, I'm not going to show much of this, but we did do RNA-seq with these buds. And we see a cytoskeleton reorganization is upregulated in the buds that gain hardiness. And actually ABA was downregulated, which is something that is a little bit counterintuitive, but we have a hypothesis about it. And I won't have time to talk about it, but if you're curious, we can talk later. And just showing some results from, this was last year, so this was 2 years ago. So last year we decided to try different temperature amplitudes. So you'll see here that if you have the buds at 7 plus and minus 3 Celsius, so going from 4 to 10, and 7 plus and minus 5, so going from 2 all the way up to 12, you'll see that when you have a range that is smaller, it doesn't make the buds gain hardiness. So comparing the blue and the orange line. But when you give it that nice 10 degrees range of temperature variation, you actually have, I just realized that every time I was pointing, it wasn't actually pointing. So here, the 7 plus and minus 5 was really where we were seeing acclimation happening. This is to show some other data with 2 and 2 plus and minus 5. You'll see that with the 2 plus and minus 5, it is gaining a lot more hardiness. So the average temperature here is still 2 Celsius, but it's gaining a lot more hardiness. So there is information there that we have to adapt into the model. So once we take this sigma T and we plug it into the model, we are able to fix the earlier portion of it. If we use it for the whole thing, it doesn't work anymore. So we have this idea that there's different responses in one phase and in the other that we have to look at. So looking at the acclimation, the same type of thing, we take buds from the field and we cut them into single node cuttings and we put them in growth chambers at different temperature to see how they are responding to temperature. This we did at multiple times during the year, so when they had different chill accumulations. And you'll see here the closed dots are at the lowest chill accumulation. So both at 10 and at 22 degrees C, they have a very negligible rate of deacclimation. So they're losing hardiness very slowly at that point. Once you give more chilling, so the open dots, 800 hours, you'll see that they start losing hardiness slightly faster. And when you give them 1,600 hours of chilling, that rate is much, much faster. And you'll see that it's not really a matter of where they are in their initial LTE, because these two time points, they were at about the same initial LTE, but the rates are very different. And so if we look at how these buds are responding, essentially if you take them from the field at different points in time, they are losing their hardiness faster and faster. And so when we look at how this relationship is, regardless of the temperature, so we're normalizing by temperature and seeing from a percentage-wise what is happening there, we see this logistic curve in the rates of deacclimation. So at very low chilling, you have a very low rate of deacclimation compared to when you actually have accumulated chill here that we call the 100% of the rate of response. And we call this a potential, deacclimation potential, because it's increasing over time until you get to 100%, and then your rate is not changing anymore. So going back there, we see that this is increasing, and it's a logistic increase. And we created this analogy for how the response happens. That is, you have your chill accumulation, and imagine that these buds are airplanes, and so when they have very little chill accumulation, they're taking off, so they're very slow, and then as they're gaining altitude, they are increasing the speed until they reach a cruising speed when it's their maximum chilling. But this really depends on what temperature they are experiencing, and are they experiencing temperatures like this in the field? So if you look at their responses to temperature as you at a point in your chill accumulation, you'll see that low temperatures deacclimate slower than higher temperatures. So essentially, you have different rates of deacclimation. So basically, you have different types of airplanes when you're looking at different temperatures, and all of them are going to respond in the same way to chilling. If you are taking off, you are slow regardless of what airplane you are. As you start gaining height, you're increasing your speed, and then the faster airplane is going to be much faster in the end. But they were at very similar speeds in the beginning. So there you go. Full speed depends on the airplane, which means that your rate of deacclimation depends on the temperature that you're experiencing. Now, when we look at these responses to temperature, you'll see that it appears to be that you could fit a linear rate in here, but it would not give us the full information that we want. So if we look at this process and think about it as a biological process, and we do an Arrhenius plot, which is this in the right, you'll see that there's a discontinuity in the process. So it's not all an exponential response like it is in the lower temperatures. The temperatures here are flipped, by the way. And so we really have to explore this process, creating different responses for low and for high temperatures. So what we came up with is that the low temperatures have an exponential response, and the higher temperatures have a logarithmic response. And combined with the logistic response for the potential for the acclimation. So really what we're seeing here is that you have at the highest chill accumulation, you have the highest potential rate of deacclimation over there. And as you decrease your chilling accumulation, you still have a similar curve or a similar response, but it's a lower rate at a lower rate. And then when you get to the very low chill accumulations here, you'll see that the rate at 22 is it's negligible compared to what it is. You're losing 0.1 degrees per day, whereas at full chilling, you're up in the 2 degrees C per day range. So we take this and we go back to our field cold hardiness, And what we're putting in there is we're integrating the effects of the rate of deacclimation and the chill accumulation throughout the whole winter. And when we plug it into the model corrected with the LTEs there, we see that we have fixed this model. And now we can predict much better where the cold hardiness is. So this was one of the objectives that we had to fix this model. So I think we're very close to getting there and having something that is useful for the farmers. But there's some more exploration that we can get from this data. So if we look at this, these are 3 different genotypes. We're looking at riparia, Riesling, and Cabernet Sauvignon. And so in the bottom you have the LTEs, so the hardiness being lost over time here. And on top we have the phenology, so early bud phenology or Budbreak, in which the 3 is what we consider to be Budbreak. And so you'll see that these genotypes, they are staggered. So the riparia loses its hardiness much faster than the others, and it also breaks bud faster than the other genotypes. And then you have Riesling and Cab Sauv. But if we take what we learned about the rates of deacclimation, and we know that riparia is faster than Riesling, which is also faster than Cab Sauv, we can fix them based on thermal time calculation for this. And so we see that we can actually put all of them together based on this thermal time. It's just the fact that they perceive temperatures differently across these genotypes. But when we look at their phenology, you see that Riesling and Cab Sauv are now together. So they are breaking bud at the same time with the same thermal time. But riparia is still breaking bud faster, which might not make much sense now, but I'll show you some data later on that explains this. So I mentioned endodormancy and ecodormancy, and I showed you how the rates of deacclimation change in that period, and that this is really is a quantitative trait. So you have a it's a continuous response in there. But typically what we do is Budbreak assays to measure the switch from endodormancy to ecodormancy. And the way that that's done is we take buds from the field and we put them into a chamber, usually at 4C, so that 4 Celsius, so they are accumulating chill at a certain rate. And then we take buds from that growth chamber into 22C, and we see how long they take to bud break. And usually the response that you see is there's differences between insufficient chilling and full chilling, and how long it takes for bud break to happen and how synchronized that bud break happens. And so you can see here with insufficient chilling that you have, you're not synchronized. So some buds are breaking well, whereas others are not. And then when you get full chilling, that bud break happens very synchronously. What this data looks like when you put it in a graph is that as you accumulate chill, here is time to bud break from when you started your assay. At low chill accumulations, it takes a lot longer, and you see that this curve is more slanted. So the synchronicity of those buds breaking is lower than when you accumulate more chilling. But really, what's important here is that the time difference from what is used for this type of data is the time to reach 50% bud break. If we look at those data there in the graph, the typical exploration of this is that we'll create a linear rate for those buds for the time to bud break. And there's an arbitrary time that is 28 days to bud break. And that's what your chilling accumulation is for that specific genotype. But when we do that, we're really ignoring curvature that you can see in this data that is there. And I'll show you how that curvature is actually similar to what we're seeing in our rates of deacclimation. The way I actually started looking at this was because when I looked at the bottom portion of that curve, that rate that is in there is actually very close to the rate of deacclimation at 4 Celsius that we saw when we were trying to deacclimate them at that temperature. And I'll show you how that affects your time to bud break. I'll skip that. So if up here we have, similar to the graphs that I was showing you earlier, we have bud break on top and your initial LT in the bottom. When you take those buds and you put them into forcing conditions, so at a high temperature, you measure their LTE there, they have a normal distribution, and they're gonna lose hardiness. And imagine that you're keeping your error there, but now you're looking at the error in the horizontal direction. So the buds that have the least hardiness are getting to the bud break part faster, and the ones that are lower are getting to the bud break later. And so if we look at the shape of the curve that we had before, it just changes from a probability density function into a cumulative density function. But then you see that, imagine that you have lower chill accumulation there, so you have a slower rate, and you have that more slanted curve. And you're taking more time to reach bud break. As you accumulate chill, so with a lot more chill accumulated in that 4C chamber, you'll see that the rates are faster. So your bud break is happening in a shorter period of time. So you see that that curve is sharper there. And when we look at the time to bud break, it's the size of the bar. So when you started the bud break assay until when you saw 50% bud break. And so you see that the time to bud break is a lot shorter. What we expect, however, is that instead of having this sort of behavior that we're measuring here, is that we are losing hardiness at 4C as you accumulate chilling. So imagine that you took those buds from the field very early in the winter and they haven't accumulated any chill. And so as you start giving them chilling, they actually start responding even to those temperatures in which they're accumulating chill. So they are gonna start losing hardiness. So instead of measuring your time to bud break as these bars that we had previously, because you're starting from a different start point, you're actually gonna have a shorter time to bud break there than what you actually expected it to be if you have the same initial LT. And so if you actually try it later on and you have already maximized your rate of deacclimation, you're still going to perceive a shorter time to bud break, but that's not based on the response of the buds to that forcing assay, but really how they are responding to the temperature in which you're storing them. So if we take that into account, then we're able to separate this in 3 different portions that you're getting to with the bud break assays, but you're combining all of them together. One of them is your initial LT. So depending where you were in the beginning of when you collected your buds, that gives you part of the time to bud break. The other portion is the rates of deacclimation. So how fast a given genotype is, that's going to give you a piece of information. And then the last piece of information is really its response to chilling. So is it responding to that chilling accumulation? When is it maximized? So that's the third piece of information that you actually combine all of those into a single data point when you explore them as the time to bud break in a linear way. So here is just showing what we expect is happening. And this data sort of works with, if you see the open dots here that are slightly lower than the closed dots, those had a higher initial LTE, so they are the dotted line here, and the dark dots are the dashed line. So it works with the data. We still have to have an experiment where we actually track the LTs in the beginning of the experiment and the time to bud break, so we can really make sure of what's happening there. But this is the hypothesis that we're working on now. I'll skip this. We looked at gene expression of buds losing hardiness, and one of the things that we saw was that the ABA pathway was downregulated. So they were decreasing their ABA production as they lost hardiness. And so we tried to use that information into actually applying into the field. So we took buds and we put them in a growth chamber. We applied ABA to them. We sprayed them with very high rates compared to what you typically have in field applications. We used 0, 10, and 20 millimolars. And what we saw was that in those that we applied ABA, they stopped losing hardiness, whereas when you don't apply it, it's still losing hardiness. And we see that also in the phenology. You have a delayed phenology when you apply ABA to the buds. So we took that information from our RNA-seq data into applying it in the field. But all of this is just taking that information, is really exploring that LTE data to its maximum. But there was this guy who said in 1897, he said that the direct observation of freezing, of the freezing cell is the best means of obtaining information on the causes of death by freezing. And that is really because we are very visual creatures. We have to see what's happening so that we really believe. So we started using X-ray phase contrast, which is a non-destructive method to look at how freezing was happening within the bud. So really what this is that we're taking X-rays and we're keeping only the portion that is very all of the X-rays that are not deviating from the regular horizontal path. We expand them and we pass them through the bud, and those X-rays will interact with the densities, with the different densities in the bud. And as they go by, you'll see that some of them are going to interact. They change their path slightly. And because of that interaction, you actually have a signal in an image. So you get your X-rays there. This is different from what absorption X-ray is when you break a bone, because the variations in density are very slight here in the green tissue. So we're not able to use the absorption X-ray. And we're also doing this while we were freezing the buds and rotating them. So we are doing a CT scan of buds at the synchrotron. And this is just to show you what it looks like. So this is a bud of riparia that we're slowly slicing through it. So this is after the reconstruction. That's what it looks like. You're seeing the xylem vessels here in the stem portion. And I think you might remember I mentioned the bud cushion. You see that there's no vascularization here in the base of the bud. So it's not really connected to the xylem. And that's what gives it the ability to supercool, the fact that it's separated from the cane. So that's what the images look like, and we can look at them afterwards. So also incorporated in this is we use needle thermocouples. They have a tiny thermocouple in the tip of that needle that measures the temperature, and we're able to stick those inside the bud. And this is what they look like. They're very tiny. And this is just comparing in the field what it looks like. So you see that the bud temperature sometimes is different than the air temperature. Now I'm showing you this video, and I'm actually going to scroll through it because you're going to see the bud. This is a bud of Amurensis as it's freezing. And what we see is really an expansion of the tissue. So I'll go back and forth, and you'll see that it looks like it's breathing. And from there, we can identify that the base of the bud is where the freezing actually starts. And then it propagates throughout the whole bud. And you'll see that the top portions of the bud is only freezing much later on. The whole process of freezing actually happens through a time span of 3 minutes in this case. And we were lowering the temperature at 50 degrees Celsius per hour. So it was a very steep rate, and it was still freezing only, it was still taking 3 minutes for it to fully freeze. So after we reconstruct these buds, we're able to see what they look like from the inside. And then you'll see this is riparia. And you'll see that the bud is really expanding there. And one thing to notice, if you remember that riparia had a different phenology compared to the viniferas, you'll see here that at day 6, you already have these scales separated. So you're already at a different state. This is stage 1, this is stage 2, and this is what we call stage 3. And you'll see that the inflorescence is expanding very quickly throughout that time. Because this is a non-destructive method, we're able to actually measure the freezing of these specific buds with those needle thermocouples. And so we see that that bud at day 0 was freezing at -22 Celsius. Then at day 6, that bud was freezing at -15 Celsius. And at day 13, that bud that was already very large was freezing at about -8 Celsius. So you see that hardiness being lost, and we're able to actually measure from the specific buds that we're imaging. In vinifera here, looking at day 0 and day 13, so the extremes that I showed you for riparia, you'll see that vinifera is still closed at day 13. Even though it's lost all of its hardiness, it's still a closed bud. You can see that it's swollen, it's a little bit larger, but we don't perceive it yet as being a stage 2. So its phenology is delayed compared to that of riparia. And that's largely because of the difference in growth within those, between those species. Vinifera is actually expanding the base a lot more than its inflorescences. So the inflorescences are still small, and it already lost all of its hardiness, as compared to in riparia, where we're seeing that really large inflorescence. Amurensis, a different wild species, is very interesting in its structure, because even at day 0, you see that there's a lot of tissue in there. So it's all green tissue. It doesn't have all that airspace that we were seeing compared to the other species. So here is looking at day 0 from those 3 species, and you'll see that Amurensis is really full of green tissue. There's no air pockets in it, whereas the other species have air pockets. They also have very different sizes. These are to scale, these images. So it was interesting to see that this was the bud that was freezing in the first video. So you can see the different portions of it expanding. So now we're able to take that information to study specific tissues of the bud and actually learn what is happening in those tissues that it's failing for its cold hardiness. So some of the conclusions that we take from the data that I showed you is that acclimation and deacclimation portions, they respond differently to the temperature stimulus. And really is the acclimation is responding both to the absolute temperature that the buds are experiencing and the amplitude of that temperature. Whereas the deacclimation is really only responding to the absolute temperature. But that's not a linear response as typically is used with those data. The dormancy transition is a quantitative trait, and this is very helpful for us because now we can explore it better, and we can separate those 3 traits that are associated with this, with the dormancy studies. And so there's information that we get from the Budbreak data that perhaps we won't necessarily see if we just use the rates of deacclimation. So perhaps combining those 2 is the necessary step that we need to really understand what is the dormancy and how is chilling being accumulated in the buds. And here, deacclimation is partially growth, and I put that partially in there because we see, especially in the field, we see reacclimation. So if they experience warm temperatures and then the temperature lowers again, they'll gain a little bit of hardiness. So it is a slightly reversible process. And if it was only growth, we know that the definition of growth tells us that it is an irreversible process. So the loss of hardiness can't be only growth, because otherwise it wouldn't, we wouldn't see that reversibility. And potentially, because it is partially growth, is that when we use ABA, we're able to stop that growth from happening, and then we prevent that loss of hardiness. So this is where the application in the field is. We might be able to, in the future, apply ABA and prevent damage in the later winter if they don't lose their hardiness. So with that, I want to thank all the people that helped me with collecting data, especially Dr. Thorne from my committee, and Ken at Chess, who is the one that pushed me into getting those images. And really special thanks for the lab group, the Reich group, and Jason. So Jason was the one who said, every time I came up with a good idea, with an idea, he would say, yeah, we should try that. And then Bruce was the one who put the steps on the brake and said, about this time last year, he was like, maybe you shouldn't be sitting on this mountain of data, and you should start working on it towards finishing your degree. So thanks for them for working together into something that I guess worked. And thank you for listening to me, and I can take questions that you may have. Okay, thank you.
Now, how practical and economically feasible do you think it would be to spray ABA on an orchard?
All right. I guess Geneva can usually hear the question, but the question was how practical and feasible would it be to apply ABA to the orchard? So that's a very good question. It is a very expensive product right now, and the concentrations that we were using were very high. But if you think about the amount of wood that is going to be left in your vineyard, and that is very small, the volume that you would be applying if it was directed would be very, very small. Of course, with that comes a lot of problems. Like the ABA is actually a photosensitive molecule, the ABA that is used. And so I'm trying it right now in the field to see if it actually works with just spraying them, because it might get degraded before it can have an effect on the buds. So I think now that we understand that ABA can have an effect and that we can use it, then we start exploring how applicable it is to use it in the field. But it's coming to we have an idea of how to mitigate the damage, not just know that damage happened. Yes.
So we'd exchanged an email about the molecular mechanisms of cold acclimation and these late embryogenesis proteins that are intrinsically disordered, and how they might act as cryoprotectants. Have you given any more thought to that, or in general to the molecular mechanisms by which they're becoming cold hardy?
Okay, so the question is if I have given any thought to the molecular mechanisms of acclimation. So as I mentioned before, we have a data set for RNA-seq on what is changed and what genes are being differentially expressed. I haven't really analyzed it fully to see what's happening. As I mentioned, there's reorganization of the cytoskeleton, which is something that that has been seen in other plants. And there's also a low temperature correlated protein that I remember seeing that is a gibberellin related response, so plant hormones playing a role. But from the cytoskeleton side, it's interesting that they are changing their structure, the structures of the cells, from a physical standpoint, because they are able to resist a very low temperature and maintaining that water in the liquid state in an interface with ice. they are actually lowering their water potential to a very negative state. So they're withstanding minus 250 bars or something close to that in those cells, which is something that doesn't typically happen except for in the winter. So you see that there's a need for structural reconfiguration of the cells. But yeah, more than that, we haven't seen, and we actually expect that a lot of it is a response to dehydration, because those buds actually lose water in a way throughout the earlier portions of the winter.
But it'd be interesting to look to see how many of the upregulated proteins are intrinsically disordered.
Mm.
That would be
The structure of the proteins, yeah.
Which ones are known to be intrinsically disordered?
Mm-hmm.
We have time for one more question.
Right, one more. Hannah.
Trying to just contextualize this within an applied plant breeding program. So, like,
Yeah.
what did you say to Bruce that he should begin selecting on to increase cold hardiness?
Right, so the question is, now with this information, how do we apply this into a breeding program? I think one of the best pieces of information that we can take from this is that split from the Budbreak assay, that we're usually looking for things that require more chilling so that they break bud later and they're not damaged by late frosts. And really, we can now split it into what has the lowest LT, what has a low deacclimation rate, and what has a higher chilling requirement. So we can really maximize that whole process to have the plants coming out later in the spring, so that they don't suffer any damage from late frost and really late winter dips in temperature after they would expect to have deacclimated. So I think really what I expected to give him were markers. What I gave him was now a phenotype so that he can go after the markers.
Thank you so much, Al.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.